So now we move into the keynote session of this meeting. We're very privileged and honored to have as our keynote speaker, Professor Dame Sarah Gilbert, who I've known for many years. It's hard to encapsulate in a brief introduction Sarah's contribution to vaccines over the last few years, but I'll try and be very succinct and brief. She is the Lakshmi Mittal and Family Professor of Vaccinology at the University of Oxford and a co-founder of Vaccitech. She is internationally renowned for her pioneering work in vaccine development against emerging infectious diseases, including influenza, MERS, and most notably COVID-19. She led the development of the Oxford AstraZeneca COVID-19 vaccine during the pandemic. Many of us in this country didn't have this vaccine here, but I know many in other parts of the world, including my parents in Sri Lanka, were the beneficiaries of your vaccine, Sarah. So your vaccine has saved hundreds of millions of lives throughout the world. And for this, we're truly grateful to you and your team at Oxford. And beyond Sarah's scientific achievements, she has also gained somewhat of a notoriety in the wider public domain. She received a standing ovation at Wimbledon during one of the finals of the Wimbledon tennis championship with a much bigger crowd than we have here. She has a Barbie doll named after her. And so these are amazing accomplishments, even as equal as maybe making a COVID vaccine that saves hundreds of millions of lives, okay? So for her groundbreaking work, she was appointed as the Dame Commander of the Order of the British Empire and elected as a Fellow of the Royal Society. So Sarah, welcome, and please join me in welcoming Dame Sarah Gilbert.
Thank you, Bali. So that's a wonderful introduction. Question the accuracy of some of it, but we can talk about that later. So I'm going to go back to the first slide in my presentation to begin with. Okay, so I've been working in vaccine development for about 30 years now by accident. I originally started working in biotech, developing recombinant human blood proteins in yeast. We were making an artificial blood. I was working on the hemoglobin, which was really cool because when it worked, the yeast went red. And we were doing really well with this biotech company, but then the parent company sold it. And as can happen, research really became less and less interesting as the purse strings became tighter. So I looked to move back into doing something more interesting. And I moved to Oxford University, making a major leap to work on the genetics of host parasite interactions in malaria, which is very different from what I'd done before. But, you know, if you can solve problems in science in one field, you can solve problems in science in another field. So I was going and give it a go. And when the work I did in malaria and human genetics led quite quickly to the discovery that there was an opportunity to develop vaccines against malaria by making a T-cell response against the liver stage, which is a short stage in the life cycle of the malaria parasite, but it's one that we could attack with T cells. Now, the malaria vaccine that eventually got licensed, R21, which was licensed quite recently, was not that vaccine. It's an antibody-inducing vaccine, another one that I also worked on. But during my time at Oxford, I moved away from working on malaria vaccines into pandemic flu vaccines, not really seasonal flu, but pandemic flu. And that took me into the wider field of outbreak pathogen vaccine development. And so I ended up working on vaccine technologies that we could use to make lots of different vaccines to deal with outbreaks. And so I'm going to tell you some of the things I've learned and how the field has changed over the time that I've been working on it. So an event that really changed things in thinking about outbreak pathogens was the Ebola outbreak in West Africa in 2014. Now, this was definitely not the first time this virus had been identified infecting humans. That first happened in 1976, and there have been multiple small outbreaks in Central and West Africa, more or less every year since then. And there hadn't been very much effort put into developing medical countermeasures, so vaccines, diagnostics, therapeutics, these outbreaks, they were small, they were dealt with by contact tracing and quarantine, very standard public health measures. They were usually in rural areas. The disease is carried in bats. Those bats don't tend to come into cities. So the outbreaks would be in rural areas, little human-to-human contact, quite easy to do the contact tracing, identify the people who've been in contact with an infected person, quarantine them. If you find all of them, then the outbreak ends and that's it. And that's really all that was done for many years. But in 2014, this outbreak was not contained. It spread. The international response didn't really kick in until the middle of 2014, when really the rest of the world began to feel threatened by this virus. So what had happened was that the initial outbreak wasn't contained. Some people with the virus had traveled to cities, higher population density, more possibility for transmission, much more difficult to do contact tracing. People were traveling when they were asymptomatic. It got into 3 different countries. It was starting to crop up in small numbers in other countries, including a traveler who came to the US and wasn't identified as being potentially infected with Ebola until rather too late, because that then resulted in transmission in this country. But those kind of events were really what stimulated the international response to try and end this. And I was one of the people in Oxford who were asked if we could please help with the first clinical trials of a vaccine that had showed promise in non-human primate challenge studies, but had never been tested in the clinic before. And so I and a team of other people got on board and we tested this vaccine very quickly. We did our safety and immunogenicity testing. We passed the information on to a group of researchers in Mali. They repeated our study. Mali was next to the outbreak area. They had some cases. They wanted their healthcare staff immunized. So that's who they recruited for their trial. And by November of 2014, we felt this vaccine was ready to go into efficacy testing in the outbreak area. And there was another vaccine, a VSV vectored vaccine. Ours was a CHAV3 vectored vaccine. There was a VSV vectored vaccine going through a similar process with different groups. Both of them could have gone into efficacy testing. We could have found out if both of these vaccines were effective. That didn't happen. The efficacy trials didn't start until April of 2015 because there was a lot of debate at this very late stage about the ethical way to conduct efficacy trials of such a pathogen with such a high case fatality rate. Now, this was a discussion that needed to be held, but in my view, it was at least 6 months too late. We should have had that protocol in place so that as soon as we had vaccines that were ready to go into efficacy testing, we had an agreed protocol to test the vaccines. But it didn't happen. It was the other way around. So we had a 5-month delay while the vaccines were ready to go into efficacy trials, but nobody was ready to do the efficacy trials. And the measures to contain the outbreak were working well. There was a lot more international effort being put in, field hospitals being built, more supportive therapy. So more people were surviving the illness because they were getting better supportive care. And the number of cases were dropping. And eventually only one vaccine, not the vaccine we'd worked on to our disappointment, but the other one went into efficacy trials and was shown to be very, very highly effective. But by the time we knew that, the outbreak was essentially over. So this was a missed opportunity. And there are many other opportunities that were missed in this outbreak. It shouldn't have been such a big outbreak, but given that it was, we should have been doing a lot more to contain it, to test medical countermeasures so that we better prepare for the next one. But this served as a wake-up call. And one of the things that happened was a number of different organizations started to come up with their lists of priority pathogens. So these are pathogens that have caused outbreaks in the past. They're likely to cause outbreaks in the future. There's no reason why that won't happen again. And we don't have medical countermeasures against any of them. And what you can see on this slide is the year when these different viruses were first identified, and on the map, the country where they were first identified, not every outbreak that's ever happened, but where they first identified. And the point of this is that it's all over the world. Where there are people, you can have disease outbreaks of a novel pathogen. So it's not predictable. And the types of viruses that we see on this list are different viral families, so that's not predictable either. Some of them are filoviruses, we've got an arenavirus, we've got bunyaviruses, henipaviruses, we've got vector-borne viruses carried by insects in some cases, with zoonotic reservoirs in bats in quite a few cases, but sometimes in rodents, and sometimes livestock are affected. And they're the amplification phase that means that when the livestock get infected, people get infected from their livestock, but that's not always true. And sometimes the livestock are pigs, so it might be sheep, cattle, or goats, or it might be camels. So there's not a lot of consistency here. So we need to do something about it. And the obvious thing to do, was felt in 2015, was to start developing the medical countermeasures against all of these pathogens, because these are going to come back. It doesn't mean there won't ever be another new one, but we already know about these. We should get on with doing something about these. A refinement of this was that disease X was then added to the list, disease X to represent the unknown pathogen. It's all very well being prepared for an Ebola, a Marburg, a Lassa outbreak. What happens if it's a novel virus? Obviously, that came to pass in 2020 when a novel coronavirus started to cause an outbreak, which became a pandemic. And the difficulty there was that knowing it's a coronavirus, we don't have vaccines against any other human coronavirus diseases. We have seasonal coronaviruses that infect us. There are 4 known. They're rarely diagnosed because they cause generally a mild respiratory illness, typically in young children who then become partially immune, but they will get infected later on in life. And if you get infected very late in life where you have a compromised immune system, you may then have a severe disease and it may then be diagnosed as a coronavirus infection. That tends to happen if there's an outbreak in a care home facility where there are a lot of elderly people with weak immune systems. You might actually then discover that what is causing these people to be severely ill is a seasonal coronavirus infection. But otherwise, it's just one of these viruses that infects us and we get over it and we don't even really recognize that it's happened. So given that we don't have a vaccine against a coronavirus, we don't really know what we have to do to develop a vaccine against another coronavirus. For measles, if you have a very low neutralizing antibody titer, you will have good protection. For yellow fever virus, a very low neutralizing antibody titer is protective because those viruses replicate fairly slowly. We don't know what's true of a coronavirus. So what do we have to achieve? So that led to needing to test lots of different types of vaccines, fortunately, most of which turned out to be highly effective and useful, but we didn't know that when we started. So there was a lot of effort put into test lots of different things. So what's now recognized is that rather than just thinking about the individual pathogens of caused outbreaks in the past, we need to think about the viral families, and we can identify an exemplar from each one of those families. And this is the WHO's website that describes all of this approach. So we could use SARS-CoV-2 as an exemplar of human coronaviruses and say, well, if we know how to make vaccines against SARS-CoV-2, we've got a really good head start in making vaccines against another novel coronavirus should it arise. If we get a new henipavirus emerging, we don't have vaccines against henipaviruses for humans. We do have one for horses, and I'll tell you about that. So we should at least be working on at least one of the pathogens from each of these viral families, because that really gives us a head start if there's a novel one and we need to respond quickly. So rather than saying, well, it could be anything, we don't know, we'll just try and go fast when something happens. That's the wrong approach. We have to be working on developing vaccines against these pathogens that we know about so we can shut down outbreaks when they happen, but also gain knowledge that we could use in the future if there's another large outbreak which becomes a pandemic. And it was recognized from 2015 onwards that really we're only going to have effective response if we use vaccine platform technologies to develop these vaccines. Now, in the middle of the 20th century, many vaccines were developed directly from the pathogen that causes the illness. So the mumps virus vaccine held the record for the fastest vaccine development, 4 years, from Maurice Hilleman taking a swab of the mumps virus from his daughter's throat, taking it into the lab, passaging it for 2 years in chicken eggs and various cell lines to attenuate it, and then 2 years of clinical trials of that vaccine, largely in children's homes where there tended to be mumps outbreaks, until that vaccine was licensed. Having been tested in relatively small number of children, but it was shown to be effective and it's licensed and it's been it's still the same vaccine that we use now. So the live attenuated vaccines was one way, but you have to take an individual approach for each pathogen and work on it until you think you've got something safe enough to go into clinical trials, but still capable of inducing a protective immune response. Inactivated vaccines were the other main technology, where you would produce a large amount of your pathogen, your virus, inactivate it chemically or with heat, and then use that killed vaccine, typically along with an adjuvant, because killed vaccines tend not to induce a very strong immune response on their own. You might need multiple doses and you need something else to boost the immune response. But both of those approaches mean you have to start with the pathogen itself and work out exactly how you can safely produce a lot of it or attenuate it. And that all takes time and a lot of investment for each one. If you use platform technologies, we do the work on the platform technology, and we use that platform technology to then make lots of other vaccines. So we spend probably 90% of our time, our effort, our money on the platform, and then we're ready to go when we need to tailor it for all these different vaccines that we want to make. So a vaccine platform technology is something capable of delivering a vaccine, antigen, and inducing immune response. So adenovector vaccines that I work on, mRNA vaccines that we'll hear about next, all are both true platform technologies. They don't produce the antigen that you want the immune response until after the person's been immunized. That means that you don't have to worry about the influence of that antigen on your manufacturing because it's not there. You just have the coding sequence, whether it's DNA or RNA, it's encoded. The protein antigen itself doesn't exist until after vaccination. So protein stability is not an issue. Purification of the protein isn't an issue. It means you can standardize so many things. Your quality control panel is almost identical for each vaccine. You just have a few antigen-specific tests to do on this vaccine. So it means you can spend less money to make multiple vaccines, which we need to do for outbreak pathogen vaccines. It means you can go faster to make a new vaccine because you've already done a lot of the work. And there are some technologies such as recombinant protein or recombinant VSV, and VSV is another viral vector where you essentially make a pseudotype virus and you have your Ebola antigen covering the surface of the VSV virus. And those are what I call partial platforms because although it's a standardized approach, the actual protein antigen that you want to make will affect how you do your manufacturing, how you do your quality control. The antigen on the surface of the VSV virus affects which cells will get infected by that vaccine vector. So it's not the same every time. So you still have more work to do, even though you're using what's sometimes described as a platform technology. But DNA vaccines, viral vector vaccines, mRNA vaccines, true platform technologies, easy to turn to lots of different purposes. Not glycoconjugate vaccines, obviously, that's another story, because we can't include the glycans. So ChAdOx1 is the one that I've been working on for some time. It's a replication deficient simian adenoviral vectored vaccine. So we've taken out the E1 gene, which allows the virus to replicate in human cells. Normally that's supplied in the manufacturing cell line. It doesn't recombine with the virus because the sequences are not homologous. So we don't generate any live virus. It will go into a cell when somebody's vaccinated, it will express the antigen and it can't make any more copies of itself. So it can't spread, you can't get disseminated infection in the way that you can with a smallpox vaccine, for example, although we've now moved on to replication deficient smallpox vaccines, which are less effective but safer. So this one doesn't replicate. It's a simian adenoviral vector vaccine because we get adenovirus infections throughout life. And if you've had an adenovirus infection and your immune system has had to respond to that to the extent that it can clear the infection, you're left with immune memory and high titer neutralizing antibodies against the adenovirus. You don't get that same level of immunity induced when you give somebody a replication deficient vaccine, and I'll say more on that later. But we wanted to avoid having to work out how much effect there would be of the neutralizing antibodies from human infections, although actually it turns out that it's not a very large effect, and there were Ad5 active vaccines used in the pandemic. So before April of 2020, when we started our phase 1 trials for the coronavirus vaccine, we'd done 12 independent phase 1 studies in Oxford, vaccinated 330 different people with lots of different antigens. More broadly, simian adenoviruses had been tested in trials of over 6,000 subjects, some of them in older people as flu vaccines, some of them in infants as malaria vaccines. And we saw consistently a safe profile, very recognizable profile of adverse events. We could predict what adverse events we would see and exactly when we would see them. And it's always in the first 2 days. And strong immunogenicity even after 1 dose. But it can be boosted either as a part of a heterologous vaccination regimen or, as I will show you, as a homologous vaccination regimen. So post 2014 Ebola outbreak, the WHO conducted their R&D blueprint exercise, asking people to come forward with their platform technologies for vaccines, diagnostics, and therapeutics. And I presented ChAdOx1 as a platform technology to make outbreak pathogen vaccines. And Janssen Vaccines went through the same exercise. We decided to partner. We went through the second round of the exercise, and this technology was recommended, but that was very nice, but the WHO didn't have any money. But what's happening at the same time was that CEPI was forming, the Coalition for Epidemic Preparedness and Innovation, came out of Public Health Norway, has received investment from Many governments, also the Wellcome Trust and the Gates Foundation, put money into CEPI, and CEPI exists to develop vaccines against outbreak pathogen vaccines. Now, it's epidemic preparedness. It was then expected to take charge in the pandemic, which it wasn't really set up to do. But the first call for proposals was to develop vaccines against one or more of MERS, Nipah, and Lassa vaccines. So together with Janssen vaccines, we said, well, we'll do all three. I'd already been working on a MERS vaccine for some years. We could do Nipah, we could do Lassa. We got some money from CEPI, and we set about doing this. So a little bit about those diseases. Nipah virus is one of the really scary ones. Very high case fatality rate, up to 100% in some outbreaks. It's carried in fruit bats. It was first identified in Malaysia after infected pigs who were eating fruit contaminated by the bats. A lot of pigs got sick. People working at the pig farms got sick. It's identified as this novel virus. It was dealt with by culling all the pigs. There was some limited spread to other countries, but it didn't get very far. It is possible to transmit between humans, but it's very inefficient. And this is very good because it's part of the larger family of viruses, which contains measles virus, which is the most contagious virus we know of. And if Nipah virus developed the ability to transmit between humans in the way that measles virus can, we would really be in trouble. At the moment, most infections occur by eating contaminated fruit, drinking contaminated date palm sap, which bats have contaminated with Nipah virus. And it can be dealt with once identified without very, very high containment. But there are instances of healthcare workers going to help a family who's got some flu-like illness, and it turns out to be Nipah, and then the healthcare worker gets infected and dies as well before it's recognized that it's Nipah. So there are cases in Bangladesh every year, occasional outbreaks in Kerala. It could happen over a wide range because there are many countries which these bats spend their time in. So we don't have a vaccine against Nipah virus or any related virus, but we do have a veterinary vaccine against Hendra virus, also carried by fruit bats, very closely related, identified in Australia because the bats were contaminating the pasture where some very expensive racehorses were grazing, and some of these racehorses got sick and died, and some people got a Hendra infection from contact with the racehorses. No human-to-human transmission, as far as I'm aware. It was all from contact with the horses. And these are valuable animals, and it's easy to track where they are. They're large and they cost a lot, and people know which horses there are and where to find them. And so a vaccine was developed for the horses. Horses in this area of Australia started being vaccinated, and that has prevented any further human cases of Hendra because people don't get exposed because they're not working with infected horses. So this was a very effective solution to Hendra virus. It doesn't work for a Nipah virus because we can't vaccinate all the bats. But it does tell us that for a very close relative of the Nipah virus, and there's actually good cross-reactivity between Nipah and Hendra. If we can make a vaccine against Hendra virus, it's just a straightforward recombinant protein with adjuvant. We can make a vaccine against Nipah virus. We just need to do it, and we need to test it. And we're not going to be able to do an efficacy trial. We kind of hope we're not going to be able to do an efficacy trial because that would require a large outbreak, which would be very scary because a lot of people would die. Be a much worse situation than Ebola in 2014. So we will have to license this vaccine for emergency use based on correlates of protection. So you do challenge studies in non-human primates who've been vaccinated. You determine the antibody titer that is necessary to confer protection. And then you do phase 2 studies of your vaccine in people of different ages, in different locations, and you see if you can match that antibody titer that is protective in the non-human primates. And if you can get emergency license, so you can make a stockpile. Then you have to have your clinical trial protocol that we didn't have for Ebola as to how you're going to use this vaccine in the event of an outbreak. But we can get all the way to emergency use licensure without having to wait for an outbreak to happen. So we started developing this vaccine using Chadox1 expressing the glycoprotein antigen. We're testing 1 dose to see what would happen in an outbreak, 2 doses to see what would happen if we want to protect healthcare workers and surveillance teams over a long period of time. Maybe we give them 2 doses. The first cohort is unblinded, studied very intensely for safety, and then we go into a blinded cohort after that. But the unblinded data from the safety cohort has been looked at, and what we can see here is the response after the first dose of vaccine, getting an antibody response, including neutralizing antibodies. It's cross-reactive from Nipah Bangladesh in the vaccine to Nipah Malaysia, the strain. It's boosted by a second dose. Neutralizing antibodies are boosted. So this looks very promising. So this is now going through further development. What you may not be able to read on that slide is these neutralizing titers, IgG titers, are reported in international units per mil. Now, this is something we couldn't do in the coronavirus vaccine trials because we didn't have an international standard, and everybody was testing their vaccines and reporting results, and people were trying to compare from one to another. And you can't do that because everybody's using their own assay because it was a new virus and it was an emergency. But when we know about these pathogens, we can develop international standards, and then we can supply them to all the vaccine developers and ask them to use them in their assays. And then we can compare the outputs from one trial and another trial because we're using the same standardized assay. So we should be developing antibody standards for each disease because this makes vaccine development much easier when we can compare what the outputs from different vaccines. So Lassa fever virus is another one we started making a vaccine against. This is another West African problem, this time carried in small rodents, this multi-mammate mouse that you can see at the bottom of the slide here. It's a very shy rodent, but it likes to get into people's houses. If you have a rodent secure house, you're much less at risk of Lassa fever infection. It gets into food stores, it contaminates them. These rodents can live with an ongoing Lassa infection for many, many months, and they shed the virus in their feces and urine, and it contaminates food stores. And when people sweep the dust up, you breathe in the dust and people can become infected. Most transmission is actually rodent to human. It can transmit human to human, and that happens in healthcare settings, and healthcare workers have died because of if a pregnant woman is infected, the only treatment is a cesarean section, and that will release large quantities of high titer virally infected blood, and the healthcare workers, if they don't have adequate protection, may be infected themselves, and that has happened a few times. So we'd also like neutralizing antibodies to try to deal with that situation. An unpleasant side effect of this is because it infects the auditory nerves, and then the T cells that respond to the virus will then kill those infected cells. You can be left with permanent deafness, even quite a mild case of the disease. So there are many children who are left deaf because they've had a Lassa fever infection. This is another public health problem. Then sometimes it's temporary, but half the time it's permanent. So many, many people infected. It's now year-round. It's not really an outbreak pathogen. It's endemic in these countries. This is an interesting one, though, because the immunology of this viral infection is different. Neutralizing antibodies are not involved in protection. They're not involved in recovery. You don't see neutralizing antibodies being induced until some months after somebody's recovered from this Lassa infection. The vaccines that are being tested are protective in non-human primates, but they don't induce neutralizing antibodies, and the binding antibody titers are all over the place, but all the monkeys are protected. They have a consistent T cell response. So we don't know whether it's a low level binding antibody response that's important, or whether it's the T cell response, but it is definitely not neutralizing antibodies. And this makes it very difficult because we want to be able to license vaccines based on correlates of protection. Everybody's happy with neutralizing antibodies as a mechanistic correlate of protection, but we don't get them in Lassa fever virus. Now, another thing about Lassa is there are 7 different lineages, not just strains, but 7 different lineages. And currently, the immune responses that we get after vaccination, both the binding antibodies and the T cell responses, are very cross-reactive, and they're cross-protective in rodent challenge studies. You can immunize with lineage 4 and get protection against lineage 2 and lineage 3. There is a move now to try to modify the antigen so that we get neutralizing antibodies, because this would make vaccine licensure easier. What it may also do is lose that cross-protective immune response, because as you'll be very much aware of from the SARS-2 pandemic, neutralizing antibodies tend to be very specific for a particular strain. And if the strain changes, you can lose the efficacy of your vaccine that's inducing a certain neutralizing antibody response. So what we wouldn't want to see is that we manage to induce neutralizing antibodies against Lassa fever virus, but they only work against one strain from one lineage, and then we have to have a whole host of different vaccines to try to cover with the 7 lineages that we already know are out there. And there could be more if we really went and looked. So this is a difficult one. So we need a better understanding of immune protection for each disease. It's not just always neutralizing antibodies. And we do need to think about cross-strain protection and how many different strains, lineages, clades, whatever the nomenclature is, we have out there, because developing something that just works against one thing is not really going to be very helpful. Middle East respiratory coronavirus is the other one of the three that we first started working on. Now, this has caused more than 2,250 cases of severe acute respiratory disease. It's another coronavirus, which you'll be familiar with now. Camels are the source of zoonotic infection. Probably not originally, it was probably originally in bats. Bats in Africa had a precursor of the MERS coronavirus, which got transmitted to camels as trade between the Horn of Africa and the Middle East in camels. Some infected camels went to the Middle East, and now it's endemic in the camels in the Middle East. And it's very much like a human seasonal coronavirus infection in that the young animals all get infected. There will be outbreaks of this disease in the camel herds where the young animals get infected. It's not a serious illness. They recover very quickly. They're only partially immune, though they can get infected later on in life. So the adults can also get infected, and people get infected from contact with infected camels. But only half the cases of MERS coronavirus can be traced to contact with a camel. But we also know that people who work with camels are often seropositive for MERS virus. So what we're seeing is asymptomatic or mildly symptomatic cases occurring in people who work with camels. They don't report it as severe illness because it isn't a severe illness. It's much like a human seasonal coronavirus infection. They develop antibodies so we can tell they were infected, but they may pass the disease to somebody else. They may pass it on to family members. And in some years ago, there was a businessman who traveled from South Korea to the Middle East and then back again. And that one person who got infected with MERS caused a large outbreak in South Korea, which wiped $8 billion off the South Korean economy from one person traveling. And he was well enough to travel, and he went to different hospitals in South Korea and spread the infection. And once it gets into hospitals, it spreads really fast. Most of the outbreaks are in hospitals. The case fatality rate there is reported as 36%, but that is the hospital case fatality rate. And it completely ignores the fact that there are many asymptomatic or very mildly symptomatic infections in the community, which are not recognized, so they're not counted. So we don't really know how serious it is. So we do need a better understanding of disease characteristics for each disease, because prior to 2020, MERS was talked about as, oh, it has a high case fatality rate, but it only affects older people. It's not actually true. It affects everybody. It just doesn't cause serious illness in most people. And it's the hospitalized people where you have the high case fatality rate. So, we should develop a better understanding of what's actually going on if we want to be able to protect ourselves against a disease. And in this case, we should just vaccinate the camels. It worked for Hendra in horses in Australia. We should eradicate it in the camels. It hasn't been there forever. This happened within the last 50 or 60 years. People have lived very, very closely with their camels in the Middle East for decades, for centuries, and found it very difficult to accept that MERS, the zoonotic reservoir for MERS, was the camels, because this has not always been the case, but now it is the case. And we could reverse that situation if we get rid of it in the camels. Again, people aren't at risk. But there is no incentive to vaccinate the camels, and it's not happening. So we'd done trials of our Challex-1 MERS vaccine some years ago with some UK funding. We'd looked at dose response here. So this is the IgG titer at different doses. You can see the dose response at the peak titer. It's maintained well a year after vaccination. The T cell response doesn't really correlate with the dose, but it's not zero on the day of vaccination, because there are some cross-reactive responses to the human seasonal coronavirus infections that we all get, but it's boosted by the Challex-1 immunization, just a single immunization, and it's maintained at this high level even a year later. And we get neutralizing antibodies as well. Now, I wanted to look at what happens if we do 2 doses of this vaccine. Can you get a higher response when you give a second dose? And we'd started doing that. But unfortunately, we started doing it at the end of 2019, and we hadn't got very far, and we had to stop in 2020 because all non-corona or all non-pandemic related work had to stop. Where we had got was looking at different intervals between the first dose and the second dose, either 4 weeks or 6 months, and we'd only immunized 3 people. So this is not statistically significant, but I think it's interesting data, so I'll present it in that spirit. What you can see on the red line is the mean antibody response in the people who previously received this dose of vaccine. And then the one person who got a first dose and then a second dose at 6 months makes this massive boosting response at 6 months. And in the 2 people who had a second dose at 4 weeks, again, here's the mean historical response. These 2 people actually didn't respond quite as well to the first dose, but they had a really good response after boosting. So it's not very much information, but it did actually help shape our thinking when it came to the coronavirus vaccine in 2020, because we weren't just thinking about a single dose. But we do need a better understanding of vaccination regimens, because it's not the case that you know what dose you're going to give and you know what interval you're going to give. You can get different results by using the same vaccine in different ways. It's not always the same. So moving on to what happened in 2020, I was reading the news and hearing about this outbreak in China and thought it would be flu or maybe adenovirus, and it wasn't, it was coronavirus. And we were working on a MERS coronavirus vaccine, and I knew what had worked as far as immunogenicity, at least in our MERS vaccine trials. And we have a GMP manufacturing facility where we can make a vaccine, but we've only got one clean room. So I had to make one choice. What are you going to design for this new virus? Well, the obvious thing to do is exactly the way we designed it for the MERS coronavirus, because that's worked pretty well. And we don't have the opportunity to try out 10 different things. We just have to pick something and go, because we've only got one clean room. So that's what we did. We started manufacturing the vaccine. We just have the SARS-2 spike protein instead of the MERS spike protein. We're doing non-human primate challenge studies with our collaborators at Rocky Mountain Labs. We have our own GMP manufacturing facility. We work with the MHRA to get approval for the trials. We started that trial in April of 2020. We were told by the modelers in the UK that cases in the UK were probably going to peak around late May, maybe June. We could get our trials underway by then. We'd have a good chance of demonstrating efficacy really quickly. But that wasn't what happened. The modelers were completely underestimated the amount of virus that was already in the country. And we went into lockdown in March because there was so much spread of the virus, and that had to be stopped. So we then had to do our phase 1 trials in lockdown, which was a challenge we haven't looked forward to and didn't really expect, and made things very difficult, because even from phase 1, we were doing efficacy testing. We wanted anybody who had symptoms of coronavirus infection to return to the clinical centre, to have a swab test taken, to have their symptoms monitored. And we had to make sure there was no spread of infection from those people coming back. So we put a huge amount of effort into video training and streamlining. We put 2 temporary buildings up outside the marquees from music festivals, just so we had more space. We had to distance everybody in the labs, everybody with masks on and not within 3 meters of anybody else. Very, very trying circumstances to be conducting vaccine trials in, but we did it, and we didn't have any infection spreading anywhere in the team. There was a point where if one person in your team was infected, everybody else in that team had to quarantine for 2 weeks, and that would have prevented us from doing the trials, because we would just have to stop. So we really didn't want any infections, and we didn't get any. But we had to go into much larger trials we'd expected. We expanded into Brazil and South Africa. AstraZeneca came on board as our manufacturing partner. And you know the story, by November of 2020, we finally had enough clinical trial endpoints to unblind and determine vaccine efficacy. And the vaccine was licensed for emergency use, and rollout started. So this was from the first clinical trial paper published in July of 2020, where we're looking at, in the middle, this all relates to the adverse events after vaccination. Now, we didn't use a saline placebo because we know this vaccine is quite reactogenic, and we really didn't want people to be able to guess whether they'd have the COVID vaccine or saline placebo. So we gave them a MenB vaccine, which is also quite reactogenic. So everybody had some reaction to the vaccine and they couldn't guess which one they'd had. So that was a deliberate choice. And basically, the COVID vaccine is still slightly more reactogenic, but you would only know that if you look at the data. You couldn't tell on an individual basis. And it was very much as expected. T-cell responses went up as expected. Neutralizing antibodies were induced with the first dose, but in this small group of 10 in the first trial, we put in a cohort that had a second dose at 4 weeks. And we saw that, as with MERS, we do get boosting at 4 weeks. So we get a high neutralizing antibody titer after the second dose, given 4 weeks after the first dose. So what should we do with our trial? Should we go ahead with one dose, which was how we designed it originally, or do we go for 2 doses? Well, we don't know what the correlate protection is. Nobody does. So we have to go for the higher dose. So that's what we did, which meant redesigning the trials, and then we ran into problems with vaccine supply. So if we could convert immunogenicity to efficacy for each of these diseases, if we knew the correlates of protection, we wouldn't have to do those extensive efficacy trials, and we would have been able to start vaccine rollout so much earlier. We did a lot more work looking back at the trials on the interval between 2 doses. Longer the interval, stronger the antibody response after the second dose. But one thing I want you to think about is that we really need to have defined endpoints, clinical endpoints for our trials, because unless you all use the same clinical trial endpoint, you can't compare results from different trials, same as you can't compare the antibody outputs. So as an example, out of a cohort of 11,500 people in our trials, there was 100% protection against death, 100% protection against hospitalization, 70% protection against mild disease, and 27% against asymptomatic, which we know because we took swabs from these people every week. So we knew if they're infected, even if they didn't. So which is your efficacy result? And depending on how you define mild, it could be a bit lower or a bit higher, and really small changes can make a difference there. So what we need to remember is that vaccine efficacy trials, you don't just get one number and that is set in stone as a vaccine efficacy. You're trying to find out if this vaccine is effective enough to use it. And we managed to generate lots of numbers of vaccine efficacy, but they all said, yes, it's effective enough to use it. So we got licensure in the UK, but for 2 doses, 3 months apart, because we know that gives a higher response and higher efficacy. And by March of 2021, we were starting to see the impact of both our vaccine and the Pfizer-BioNTech vaccine, which were being used in roughly equal numbers at this time in the UK. In people who'd had 1 dose, they were over 70, because this was done age de-escalation. We were starting to see 60% protection against symptomatic PCR positive disease in the over 70s. In the over 80s, hospitalization was rapidly coming down and deaths in the over 80s were coming down. So even after the first dose, we were seeing the impact, and you couldn't distinguish the impact of that. It was the same, whichever vaccine people had had. We went on to look at anti-vector immunity, and really there's no effective anti-vector immunity. We didn't see any difference in people who had neutralizing antibodies pre-vaccination, or you can give the same vaccine 3 times, and people who'd had a prior Telix vaccine still made a good response. So we don't really need to worry about anti-vector immunity, which is a question that everybody always asks me. And we found out more about dosing interval. So when's the ideal time to boost? Well, it depends what you're trying to achieve. If it's a pandemic, you want it to be a short time. But if you're trying to protect healthcare workers who might be exposed to Nipah some years in the future, you probably want the second dose at a longer interval, because you will get better protection and longer lasting protection. So again, it's not a simple answer. Equitable access across the world, really important to the University of Oxford. We are a charitable institution. Vaccine was not for profit, and we wanted it to be used wherever it could be. And because it didn't require frozen storage, that was more easy to do. And we have to remember that nobody's safe until everybody's safe. AstraZeneca did a fantastic job with setting up manufacturing facilities in 15 different countries deliberately for global supply. Because if we don't vaccinate, if we don't manufacture, we can't vaccinate. If we don't have doses, it doesn't matter how fantastic the vaccine is, it will have no impact. So we need a lot of doses, and AstraZeneca really did achieve this. So we need to learn the lessons looking back at the pandemic. People tend to come in with a very narrow focus against just one aspect of vaccines, but actually everything matters. Thermostability matters, supply matters, cost per dose matters. If you're going to have big impact in the world, you have to factor in all of these things. And in an independent analysis of deaths averted in the first year of vaccine use, it was the AstraZeneca Oxford vaccine, as it is named here. We like to say that the other way around. But this is the one that came out on top with most lives saved. And that was a feature of many different factors: thermostability, cost per dose, all of it, as well as vaccine efficacy. You have to remember all of those things feature in there. We did, unfortunately, identify a very serious, very rare adverse event, vaccine-induced thrombocytopenia with thrombosis. Only risk factor is younger age. The epidemiology is that it is more common in Northern Europe than in Central and then in Southern Europe, and rarely seen in the global South. Even in countries that have very good pharmacovigilance reporting, they weren't reporting this. They're reporting Guillain-Barré syndrome, other things that you can get after vaccination, but they weren't reporting BIT. So there really does seem to be a global distribution, and we don't know why. And this is part of the focus of my research now, to understand this. So overall, can we prevent outbreaks from becoming pandemics? Because that's what we'd really like to do. And the answer is yes, we can, but it's not a simple answer. It's not the same one size fits all. We have to, even though we're using platform technologies, we have to understand the disease characteristics, the transmission, all of the rest of that, so that we can tailor our response. And this was the 2015 MERS outbreak in South Korea. One person caused an outbreak with 36 deaths, nearly 7,000 people quarantined, and $8.5 billion loss. So we want to avoid that happening, and we should be able to do that, but we need to think about the best way to do it. There are still technology challenges for any platform. Both the adenovectors and mRNA vaccines are now undergoing iterative development. We will see improvements in the future. So there's a lot more to do, and it's a really exciting time to be a vaccinologist working in this field. If you need any further reason for doing that, if we don't, this is the level of economic loss that we saw in the pandemic. This is the impact on people being pushed into poverty when those numbers have been coming down. That is purely because of the pandemic. The 100-day mission is one approach. For me, being able to do something in 100 days means you have to have done most of it beforehand. We can't just wait and then go quickly once something new happens. We have to do much more than that. If we don't, we end up with this example of the pandemic, where at the top you can see all these different colors, these are different variants taking over one from another. This is a very complex situation that arises because we allowed this virus to spread around the world and do its own thing before we got on top of it. We don't want to see that again. So we have to be prepared to act quickly, because if we don't, things get very complicated and very expensive. Our response in Oxford was to bring all the people who worked in pandemic response together into a new institute, the Pandemic Sciences Institute. We worked on not only vaccines, but also therapeutics with the recovery trial. We have people working on diagnostics, in equitable access, and we've kept these people together, and we have a conference every year, and we try to work out how we can do better in the future, because we didn't really learn that much from Ebola 2014, and we still have a long way to go. Thank you.
Thank you so much. Bali just asked me to facilitate with questions, so please, yeah, raise your hand.
Thank you very much, Sarah. Beautiful talk, beautiful work. I had a couple of questions. So one, I was quite surprised why you don't see anti-vector antibody responses. And I was wondering if you had any thoughts on that.
So we do see anti-vector neutralizing antibody response,
Yeah.
not at a very high level, because remember that we just inject the vaccine vector. There's one cycle, well, it infects cells, there's no replication. So those proteins aren't produced in any further quantity the way they would be if you had an actual infection. And even though people have got neutralizing antibodies, it doesn't seem to affect their response to vaccination.
Yeah.
If you were to, there are some cancer vaccine protocols where people use the same vector, whether it's an adeno or pox virus, and revaccinate every 3 weeks.
Sure.
And there, after about 6 vaccinations, it becomes useless. Yeah. But that's a very intensive use of vaccines. And we don't need to do that with the outbreak pathogen vaccines. Yeah. Also, remember, these are outbreak pathogen vaccines. We're not recommending that we use the same vector to make vaccines against all the common illnesses, but we can use them to make vaccines against all the outbreak pathogens, because people aren't going to receive all of these vaccines. And our aim, which doesn't work for companies, but it works for us. Our aim is to need to vaccinate as few people as possible, because the sooner we control their outbreak, the sooner we can stop vaccination. So we're not trying to sell vaccine, we're trying to have it there and use it and stop it so that few people get vaccinated. And that it's very, very unlikely that people will receive multiple outbreak pathogen vaccines then on this same vaccine platform.
Yeah, thank you.
Thank you very much. Excellent presentation. Since many cancers present neoantigens, can you use your platform for making cancer vaccines? And if so, are you doing that?
Yeah, absolutely. I mean, the platform is being used in some cancer vaccine programs. More work's been done on the vaccines that are caused post-viral infection. So therapeutic vaccines against HBV and HPV have been in development, but also people are using it in a lung cancer vaccine program. So yeah, you can, and you get good T cell responses with adenovectors. So yeah, there's an interest in doing that.
I'm just curious about the vector immunity, I think, follow up on Bali. 2 doses, clearly you got a booster to the vectored antigen. What about 3? And what about how long you have to wait after someone's received 2 doses to have that dose work effectively, let's say, against a different antigen? Has any of that information become? There
is some information. If I just get quickly back to So this slide in the center, so this is 3 doses of the same vaccine in the pandemic. So this is the first vaccination. This is a log scale for the antibodies. So here's your response after the first vaccination, boost it, it goes up again, boost it, it goes up again. This is 3 doses. And in Brazil, they gave 4 doses. But
All
that
the
was
same amount.
exactly the same.
Yeah.
In Brazil, it was over the course of about a year. So I think the first 2 doses were 4 weeks apart. And then a few months later, they gave another one. A few months later, they gave another one. It's a boosting every time. It's better to leave a longer, there's no hard and fast rule. A longer interval, you have even less impact of anti-vector immunity. But yes, you can use the same vaccine multiple times in the
So
same person.
that's the basis for your optimism on reusing it.
Yeah.
That's
Yeah.
good. Thanks.
Oh, and here this is people who'd previously been in a Challux vaccine trial with a different antigen. And then we asked them to come into the COVID vaccine trial. So there's a slight difference after the first vaccination, but these error bars overlap and then they're the same response at boost.
You don't really think there's a need for a different modified vector, a different?
We do
Okay.
have 2,
Okay.
and we can develop more. But for outbreak pathogens,
With what characteristics, I guess?
well, just with different coat proteins. The capsid is slightly different serotypes.
Sarah, if I could ask one final question. So to follow up on the information that you laid out for Nipah virus, which is terrifying, obviously, if there were to be an outbreak, could you just review what the bottlenecks would be and sort of what the timeline might be for vaccine?
Yeah, so the nightmare scenario with Nipah outbreak is that there's some large social event where people from different parts of the subcontinent come to, and there's some contaminated fruit and people get infected and then move apart again and take the infection with them, because you don't become seriously ill for several days. So then you could see outbreaks happening all over the place. But if it's contained in one area, what you'd want is to have a vaccine stockpile and to have a protocol ready to roll out that vaccine. And then you just start doing ring vaccination. So you identify the cases, you vaccinate everybody who's a contact or a contact of contacts of the cases, and then you contain the outbreak in that way. Now in Bangladesh, which is where most of the outbreaks happen, and they're very small outbreaks, they have very good surveillance. It's in the north of Bangladesh. It's in a region that's about 1/3 of the country. And if somebody has an illness that looks like Nipah virus infection and there's some evidence they may have consumed contaminated date palm sap or fruit, then the surveillance team will go and investigate, and they do a very good job. So they tend to contain it quickly. But when it happens in India, because it happens more rarely, they don't have the same surveillance plans in place, and so it spreads. So it really depends where it happens. There's lots of other countries where you could have an outbreak. The vaccine's currently being manufactured by Serum Institute of India, who are one of the SARS-2 vaccine manufacturers. We're just starting phase 2 trials in Bangladesh with that vaccine now. There is quite a large amount of vaccine available. So if an outbreak happened today, we have stocks of vaccine. We'd need to make emergency arrangements to get that vaccine to the outbreak area, but we would be able to vaccinate and contain the outbreak that way.
That's great. Thank you. Any final? Oh, good.
Beautiful talk. Question about the Lassa virus. If I understood correctly, you said that basically neutralizing antibodies are produced only very late after infection. Is it the mechanism known? Is it because it's a cytokine environment that is suppressive for B cell responses, or does the virus infect and kill the B cells?
Well, we don't really know. It's a very heavily glycosylated protein, which probably has a lot to do with it. Now, monoclonal neutralizing antibodies are protective. If you make monoclonals and you do challenge studies in non-human primates, they can protect, but you just rarely induce them either with infection or with vaccination. So there's a lot of work on redesigning the antigen, which on paper or on the computer looks fantastic. But actually then when you start immunizing animals, you still don't see neutralizing antibodies as yet. So we haven't cracked that problem.
Thank you so much. Beautiful talk. Thanks.